Let's get started. This is the session, a poster discussion session called Getting to the Heart of Immune Activation. To my right is my co-moderator, Müller-Trutwin, and I'm from Pasteur, and I'm Michael Letterman from Case Western Reserve University. What we're going to do is change the rules just a little bit. You have it listed that each talk goes for 5 minutes, one followed by the other. We're not going to do it that way. We're going to give each speaker a 5-minute time to talk. That's 4.5 minutes metric. And then after that, they will open up for about 5 minutes of questions for each specific talk. So let's get started. Our first speaker is Mary Kearney, and the title of her talk is A Higher Fraction of Drug-Resistant Proviruses Express Unspliced HIV RNA During HAART Compared to the Archival Wild-Type Proviruses that Comprise the HIV-1 Reservoir. You've got 2 minutes left.
That is a long
It's quite a title.
I apologize for that long title, but I hope I'll make it clear what it means in my talk. But the slides aren't showing on...
No, you just, excuse me.
This one. Oh, click on the link. Okay. Okay. So my talk is related to these expanded clones that have been described now by many groups. These are single infected cells that were infected prior to initiating HAART and persisted and underwent clonal expansion or cellular proliferation, so that now you have multiple populations of cells with identical proviruses. And at CROI we presented that if you look within a single expanded clone, it's about 5% of the cells that are actively infected. In other words, they're actively undergoing transcription, and if they're replication competent, probably producing virus particles that result in plasmaviremia, while greater than 90% of the cells within these clonally expanded populations are latently infected. And the fractions were not different for clones that had replication competent proviruses versus those that had defective proviruses. So we wanted to take this to the next step now and ask if HIV RNA expression in clones differs from expression in cells that are infected with proviruses that are resistant to the current HAART regimen. So to do this, we studied this patient number 1, who was in the Moldarelli and Hughes science paper in 2014, and in the Simonetti and Moldarelli PNAS paper in 2016. Sorry, that was 2014 and 2016. So this patient had multiple unscheduled treatment interruptions and ultimately failed therapy. So the last time point highlighted there in the red circle was during treatment failure. So this is a sample where the viral load was greater than 100 copies of HIV RNA per mil. So he was just starting to fail treatment. So we took that sample and did single genome sequencing on the plasma. And what we found was 2 populations in the plasma. We found a population of drug-resistant viruses. Those are shown in the open diamonds. And this is a highly diverse population, suggesting that this is likely undergoing multiple rounds of viral replication, which would make sense because these variants have multiple drug resistance mutations against the current regimen. But there was also a wild-type population, and that's shown there at the bottom of the tree in those identical sequences. So this wild-type population, every sequence is identical, likely because it's just expression from an expanded clone. So we did the integration site assay on this sample and found that about half of the integration sites were single integration sites, and half were from expanded clones. The largest of these clones was called AMBI1, and that was published in the Science and the PNAS paper. And this AMBI1 clone had a replication competent virus and matched the virus that was in the viremia of this patient, this wild type virus here. So indeed, these are wild type viruses that were being expressed from an expanded clone, while these are drug resistant viruses that are replicating in the presence of treatment. So just to repeat the question then, we wanted to know what fraction of cells with drug resistant proviruses express HIV RNA during HAART, and how does this compare to cells with wild type proviruses? So I'm going to jump just to the conclusions now, since I have just 5 minutes. I won't get into the methods of how we did this, but the paper on the methods, it's published. It's called the CARD SGS assay. It was published in PNAS this year. And using this assay, we can look at RNA in single infected cells. And so we found with the wild-type proviruses that it was about 5% of cells with wild-type proviruses that are actively infected or transcribing HIV RNA. So greater than 90% are latently infected. With the drug-resistant proviruses, it was 19%. So in the replicating population, about 19% of the cells with drug-resistant proviruses were expressing HIV RNA. So we're likely resulting in the virus that you see in the plasma. So we can look now also at the levels of RNA in single cells. And we did this with the wild-type proviruses, and we found very low levels of expression in the cells that were infected with wild-type viruses. So this is just looking now at that 5% that are expressing. And then in the drug-resistant proviruses, looking now just at that 19% that were expressing, we found some cells with these high levels of expression. So these are cells that have 30, 40, 50 copies of unspliced HIV RNA. And when we sequenced from these, and by the way, these are statistically significantly different. So when we sequenced from these high-expressing cells, we found that indeed they did match what was found in the plasma viremia. So likely it's these high-expressing cells that are resulting in the virus that you see in plasma and in virologic failure. So to conclude, the fraction and levels of proviral expression were significantly higher in cells with drug-resistant variants than those with wild-type proviruses. And these findings show that HAART can select both for cells infected before HAART initiation that either do not express HIV RNA or express at low levels, or for cells infected recently with drug-resistant viruses that express at higher levels. So I'll just show, I think I've probably gone over my 5 minutes, so I'll just show my acknowledgments and just state that most of this work was done by Andrew Musick and John Spindler in my lab.
So this presentation is now open for questions. So Mary, do you think that there were in acute infection or early on in infection cells that were expressing high levels of RNA, but then they just didn't make it to persistence and latency and persistence?
Yeah, exactly. So I think that over the duration of treatment, you'll select for cells that are latently infected, which is more than 90% of the cells, or express at very low levels, and those are the ones that survive. So the high-expressing cells, I think, die off over the course of treatment.
And among the cells expressing drug-resistant virus, you suspect some of these were very recently infected?
Exactly. So I think that small fraction of high-expressing cells are the ones that were infected likely just the day before. So.
I was wondering, so this is the analysis on a patient that is very well described. I wonder to what extent your feeling is whether you would see similar proportions in other patients, for example, those that have been infected already with drug-resistant virus. How much variability do you think you would see between individuals?
Yeah, so we've looked at a lot of individuals who don't have drug resistance, and we see similar levels of expression in the wild type in other individuals, but this is the only one we've looked at with drug resistance. Well, that's not true. We looked at a patient who had drug resistance because he had failed a prior regimen, but then he was fully suppressed for 5 years on a new regimen, and in that patient we didn't see differences in the levels of expression of the drug-resistant viruses because, of course, they were sensitive to his current regimen. So in that respect, they weren't different than the wild type. Yeah.
What do you think you might find if you had the opportunity to look at a person who was recently infected with a swarm of viruses, some of which were drug-resistant, acquired drug resistance, and some of which were not? What would you predict?
So let me tell you that, first of all, we've only looked at one recently infected patient so far and measured the levels, the fraction of the infected cells that were expressing and the levels of expression. And interestingly, in chronic infection, what we had seen is, something like 5 to 20% of all of the infected cells that are expressing. The rest are latently infected. But in an early infection, and this is an N of 1, we actually found very high levels of cells that were expressing, and we found so a high fraction and high levels in early infection, but he didn't have drug resistance, so I don't know about transmission of drug-resistant variants.
Please come up to the microphone, and if you have a question to ask. There's one right over here.
So, thank you. Can you just educate us? What is the implications of these findings to general ART care?
Okay, so I think the implications are that that it's actually just a small fraction of all of the infected cells that are even transcribing at all. So most are just latently infected. So it's a small fraction that are expressing it all. Those that are expressing, the majority of them are expressing at low levels. And it's just a very small fraction that are expressing at high levels. And those appear to be the source of viremia that you find in the blood. So the virus that you see in the blood is actually just coming from a very small fraction of all of the infected cells. And so I think that it tells us something about how these infected cells persist on ART, and that is that they're either not expressing at all or they're expressing at very low levels, so they're able to escape the immune response and viral cytopathogenesis as a result.
Last question here.
I'm just wondering, so if you give these patients drugs that are active against these viruses, resistant virus, what would happen? Do you think that the amount of DNA would suppress, or what?
So actually that was done in this individual. His regimen was changed, and when the regimen was changed, the drug-resistant virus in the plasma went away, and what remained was just the wild-type virus that came from that highly expanded clone. So we haven't looked at the RNA in the single cells at that time point, but that's a good question because actually it makes it obvious that we should do that. I don't know why I didn't think of that, but we should do that and see if those high-expressing cells go away. And my guess is that they probably go away because they die. Yeah.
Okay, thank you. So our third presentation is by Davey Smith from University of California, San Diego, and the title of his talk, a shorter talk, probably because of the neurocognition issue, is HIV reservoirs in the brain and their association with sex and neurocognition.
Thank you. So today I'm happy to present on behalf of my co-authors, and these are the funders and the people who supported us, and specifically Michele off there to the left couldn't make it here because of visa issues from the United States, and Sarah Gianella on the other end, who's around here somewhere. Okay. So our goals were to compare HIV DNA levels in post-mortem brain tissues from HIV-suppressed men and women. And we wanted to evaluate its association with pre-mortem neurocognitive functioning. And we had 63 of these individuals from the NeuroAIDS Tissue Consortium, 12 men and 51 men and 12 women. And then we did neurocognitive testing before they died and 7 neuropsych abilities at the last visit. So it's about 3 months before the death. And then we sampled the frontal lobe, the occipital cortex, the basal ganglia, and the peripheral lymph tissues, and then did the HIV DNA levels there. And we measured HIV DNA levels off a GAG and 2 LTR. And then we normalized those to the cellular input. And for the GAG levels, we found that the majority of brains, about 65%, 2/3, and all the lymph tissues had detectable levels. For 2 LTR, we had 21% of the brains had detectable levels, and only about half the lymph tissues did. Lymph tissues had higher levels of HIV DNA than brain tissues, but they did not predict the levels of, that we saw in the brain. Both the basal ganglia and the frontal cortex had higher levels of HIV GAG than the occipital region. And then women had higher levels of HIV DNA in tissues than men for GAG. And that's what this figure shows. So log on the y-axis is the log copies of DNA. The women are in the darker and then the men in the lighter circles in every region that we saw, but the spread was quite large. And then we did a multivariate analysis testing for a bunch of covariates: age, estimated date of infection, time on therapy, most recent CD4, nadir CD4. None of those were associated with HIV DNA levels. We then looked at HIV levels of GAG in the occipital region, and they were associated with worse neurocognitive functioning before death in both sexes. And that's what this figure shows. The T-score is neurocognitive functioning, and the lower the T-score, the worse performance, and then men and women on the top. There were sex differences in neurocognitive functioning in motor and speed information processing in the subdomains, with women having better functioning than the men, regardless of HIV GAG levels. And that is it.
This presentation is open to questions.
You probably said this, Davey. Is this turned on? Did you normalize for target cells, CD4 positive cells?
We
So
did-
you said per 10 to the 6 cells, but was that 10 to the 6 CD4 cells?
No.
Okay.
For the lymph, we did on a separate analysis, but not for the brain, because we couldn't get the CD4s out of the brain.
Okay, so we did a comparison just between the blood and the lymph node into a small cohort of patients and found that when we normalized for CD4, target cells, that the numbers were not different between the blood and the lymph node, but.
Yeah.
Davey, I'm just wondering how you did such a study to get neurocognitive function on 60 some odd people who then went on to die.
Yeah, so that's the magic of the study. So the NeuroAIDS Tissue Consortium has a very large cohort of individuals who they follow for long periods of time, and they do neurocognitive testing, and then some of those people die. So it's a long-term study with long thought-out process. There are, as a plug, we are having new cohorts now called the Last Gift cohort, where people who are HIV infected but are dying of something else, and then they participate before they die, and then after they die, they give us their whole bodies, and then we're able to examine the different reservoirs over time.
What incredibly valuable cohorts those are.
Well, it requires very altruistic individuals, and that's very, we're very honored to be able to participate in those studies.
So you saw a difference between females and males.
We did.
You looked at several markers. Maybe I missed it. Did you look at activation of inflammation of macrophages, glial cells, or some systemic markers of monocyte activation that might be higher in females?
We didn't, and we're going to, both looking at LPS and also immunologic markers of activation. Those are the pre-mortem samples that we're going to test.
And do you interpret the lower levels of 2-LTR circles in the brain as reflective of less viral infection at that site?
Active replication, I think that's right.
And regarding your patients again, are these patients that have been put under treatment quite late because you have so much virus in the brain that the virus, the CR was too late?
They're all chronically infected when they were placed on therapy for this cohort.
What proportion of them were virologically suppressed around the time of death or before death?
All of them. And we actually could get blood, so we tested blood samples for virus at, post-mortem, and they were all suppressed.
Since you're monopolizing the questions, just kidding. This is very nice data. Do you have co-infection history of the patients? So the ones, for example, since it's a NeuroAIDS, I presume they had a detailed history. Because infections in the CNS, for example, neurosyphilis may increase, seeding of the CNS and such.
Yes. So we have those data, and we included those in the multivariate, and we didn't see anything. Although there were very few of them none of them had neurosyphilis, by the way. But toxo was the one that we were also looking at very closely. And none of them also had any history of cryptococcal meningitis.
If there are no more questions, thank you very much. I'll now...
So the next speaker is Gabriel Duet. He's a PhD student at the Institute of Biomedical Research in Argentina.
Okay. Thank you for the invitation to speak here. So, as you know, a current problem is the chronic inflammation in HIV-infected patients treated with antiretrovirals. And we know that cell metabolism influence the functionality of immune cells. And we also know that HIF-1 alpha is a transcription factor that coordinates cell metabolism and inflammation. So our aim was to analyze the HIF activity in CD40 cells and its role in HIV-associated inflammations. So, first, we wanted to know if the HIV infection modulates the HIF activity, the HIF expression, And for that, we infected primary CD40 cells with GFP-expressing HIV, and then we sorted the GFP-positive population or replicative HIV population and the GFP negative population. And we use as a negative control mock infected cells. And interestingly, we observed that the expression of HIF-1 alpha was higher in the GFP positive populations. So we wondered if the transcriptional activity of his transcriptional factor was modulated too, and for that we generated a reporter cell line that expressed GFP under the control of HIF-1 alpha. So we infected these cells, and we saw that the transcriptional activity was modulated too. And finally, by intercellular staining and by flow cytometry, we could observe that CD40 cells from HIV patients have overexpressed HIF-1 alpha. So we asked which is the viral component responsible for this effect. So for that, we did a lot of experiments, and I'm showing here one of them. And in this particular experiment, we used an integrase deficient HIV, and this virus was able to promote the activity of HIF-1 alpha. So for result that is now shown here, we know that double-stranded DNA, the fact that we used an integrase deficient virus said that the DNA generated by the reverse transcription is enough to induce the HIF activity. So the double-stranded DNA, we know that for our result, not shown here, that have the possibility to promote the secretion of extracellular vesicles. So we wondered if these extracellular vesicles are able to promote the HIF activity in non-infected cells. And for that, we infected primary CD40 cells, and 2 days post-infection, we isolate the extracellular vesicles, and we took the soluble fraction 2, and we exposed our reporter cell lines to both. And interestingly, we observed that just the extracellular vesicles from infected cells were able to promote the HIF activity. So as I said before, HIF-1 alpha is involved in inflammation. So we cultured primary CD40 cells in presence of extracellular vesicles from infected and uninfected cells. And we saw that the CD40 cell exposed to extracellular vesicles from infected cells were able to produce more interferon gamma. And if we use an HIF inhibitor called economycin, the production of interferon gamma was significantly reduced. And similar results were observed when we measured IL-6 and IL-1 beta production by macrophages. So our result suggests that after infection, the reverse transcription produced double-stranded DNA, and this viral DNA is enough to activate the HIF-1 alpha activity, and this increased glycolysis in infected cells. I am not showing here results about that, but we have some data about this. And finally, we show that the double-stranded DNA is enough to produce more extracellular vesicles, and these vesicles have the potential to promote the HIF activity in non-infected cells and promote the secretion of cytokine pro-inflammatory in non-infected cells. So that's it all.
It's time for questions. Jake?
Thank you. I think I understand your model, and it seems to be a model of viremic infection. So I'm wondering how you envision this process in an ART-suppressed patient. And to follow up on that, if GI tract damage does lead to microbial translocation, like many of us have demonstrated, and we think that that is a source of persistent inflammation in ART-suppressed patients. Would you expect to see a similar thing if you treated your cells with microbial products? Would you see an equivalent increase in HIF-alpha expression?
Actually, it's a good question. Yes, maybe it's possible. We don't know. We don't make experiment about that, but we thought that is possible because if HIF-1 alpha is totally involved in inflammation and is activated when some stimulus that pumps activate the immune cells. I don't know. Okay.
You have shown that that the activation of HIF-1 alpha is independent of the integration event. So that of the integration event of the virus, because you were using integrase defective viruses. Yes. That means that anything of the virus before the integration event can be activating, right? Doesn't need to be the reverse transcriptase, or do we have any data that it is coming down to be the reverse transcription or anything specific in before integration?
Yes, it's a good question. Actually, we did an experiment where we inhibit the retrotranscription or the reverse transcription, and we saw that this effect was abolished, was inhibited, sorry.
Okay.
We use efavirenz, and with this drug, we inhibit the HIF activity induction. So,
okay. Thank you.
I was wondering for the DNA that is accumulating in the cytoplasm, do you think it induces inflammasome activation because you see IL-1 beta, and that could induce this whole factors that you see?
Yes, it's a good question. Actually, we didn't make this experiment, but for results of another group, we know that the cells from blood don't induce the inflammasome, and specific cells from the secondary immune tissues are able to induce the inflammasome. But these cells, no.
So in the system where you generated exosomes capable of transmitting the HIF to other cells, you used an integrase defective virus. Are the levels of DNA in that particular setting greater than in a regular infection with a wild-type virus? And can you generate the same sort of exosomal activity with non-defective virus?
Okay. Yes, that is a good question. Actually, this virus is not the reverse transcription is not complete in this virus, because this phenomenon is dependent of the integrase, and we thought that the wild-type virus, yes, is able to produce more DNA than this virus. Yes, actually we did some PCR where we measured the DNA, and we found that the DNA from the wild-type virus was more than the integrase defective virus.
I was wondering, do you need some specific requirements of the CD4 T cells to be susceptible to this phenomenon?
Yes, actually, sorry.
Yeah, you see it in certain subtypes, or according to the activation or metabolic activation?
Yes, actually, this is an in vitro model, so we activated the cells before to infect with this virus.
Okay. Thank you, Gabriel. So we close this session. I would like to thank the speakers for the interesting results and for having made the effort to synthesize that allowed us to put the questions. And I invite everybody to continue to discuss with the speakers. And have a nice day.
We'll do it again. So.
Yeah.
Oh, Fudd? No, he's gone. I didn't want to put him on the spot.
No.
But I think it's a bad idea to use TLR like that.
I mean, that's what they, in the introduction they say they want to look how you can manipulate, in the sense that they know that they can enhance infection by this.